Before that, if anyone has any doubts, please, this is the time, ask your doubts. Otherwise, who among you will volunteer for a summary? How about you, Yusuf? You first. Would you like to summarize what we did in the last class?
So we just started the chapter.
Yeah, so you have in front of you, can you see the board, right? You can see
Yes.
the notebook.
Yes,
You can take that help, and if you need any help, I'll just be there. Okay, so basically you know how the summaries work. Yesterday you saw someone gave the summary, right?
Yesterday?
Did anyone give summary yesterday?
No.
No. Okay, so let me spare you for today, and let me go to either Iram or Devangi. Anyone among two of you would like to volunteer? Iram or Devangi? Whoever says yes first can go. If no, then I'll pick one. There's still 50-50 chance, so it's better you volunteer. No? Okay, if I have to pick, let me go alphabetically. Devangi, would you like to give a quick summary of photosynthesis?
Okay, sir.
Yeah.
So,
Thank you.
photosynthesis is the physiochemical process by which plants use the light energy to synthesize organic compounds like glucose in their cells.
Yes.
And glucose formula is C6H12O6, and we obtain the carbon from carbon dioxide and hydrogen from water.
Yes.
And
where do we get the
oxygen also.
oxygen from?
Yes, from carbon dioxide.
And how do we know that it's not from water but from carbon dioxide?
So I forgot the scientist's name, but
Okay.
he conducted an experiment using hydrogen sulfide instead of water.
Yes.
And then he found out that instead of oxygen, sulfur was being released.
Yes, so he figured out that it's whatever is released, whatever is present with H2, whether it's H2O or H2S, gets released. So even if it is water, we are not using that oxygen, we are releasing it out. So the oxygen that we are using to make starch is of carbon dioxide.
So
Very
what
good.
was the name of the scientist?
Cornelius von Neil. The name of the scientist was Cornelius von Neil. Yes, continue. Then we started with some early experiments in the last class.
Yes, sir. So the first scientist was Joseph Priestley, and he was the scientist who discovered oxygen, that the element that is necessary for life is oxygen.
And how did he figure that out? Is there, Devangi?
Sorry, sir, there was some disturbance.
Yeah, it's okay.
So basically he took, he first of all lighted up a candle and placed a jar over it, and inside the jar he also put a mouse. And then he observed that after a few hours, the mouse was dead. So he concluded that there was something in the jar or in the air that the mouse required to survive, and that was being used up by the by combustion, by burning.
Perfect.
Then in the second part, he repeated the experiment, but this time he also put a plant inside the jar. And after a few hours, he observed that the mouse was not dead. Means the element which was used up by the mouse and by burning of candle was being released by the plant.
Perfect.
So he discovered that plants release the oxygen gas.
Yeah, so it took him 4 years from this observation onwards. So in 1770, he made these observations through these early experiments. And in 1774, he ended up discovering O2, oxygen as an element in the atmosphere as a gas. Okay.
Yes, sir.
So it was figured out. So first piece of the puzzle was that plants released oxygen gas. Then came Julius von Sack. Yes. Continue.
So Julius von Sack discovered in 1854 that green plants also produce a compound called starch or glucose, and starch was the first visible product of photosynthesis.
Yes, right. So we figured out two things. Oxygen is released, starch is produced. Now, what was left to figure out? The fact that water is also used and CO2 is used, and oxygen from CO2 is used, not from water, was then proved by Cornelius von Neal. So that should be the third experiment that we have already discussed. Now, there is one more that we need to discuss, and that was done by T.W. Engelmann. So let me start today's class from T.W. Engelmann. So what he figured out was that there are some green algae known as cladophora. Okay. So he used prism. Very early, like in 17th century, Newton showed that you can use prism to split the white light into 7 different colors called the spectrum, Vibgyor spectrum, which is also seen in rainbow. So you know the physics behind that, I think by now. How does 7 colors split from the white light, sunlight? Some physics 101. Am I audible, people? Is it better this way? Is my voice audible to all of you? Because you are not audible if anyone is speaking. Devangi, am I audible? Okay, I think I'm
Yes,
audible
sir.
because Aisha
Yes,
gave a
sir.
thumbs up. Okay, cool. So my question is, you know, how is all different colors of light split from the white light?
So by dispersion.
So it's called bending of light when it moves from a rarer medium to a denser medium. So prism is a denser medium. It's a glass shaped in a it has a shape, a 3-dimensional triangular shape, from which when light enters, so it moves from rarer medium to denser medium, and then it bends, because different wavelengths of light have different velocities in denser medium. So all the components of white light split away into 7 different lights. Now T. Engelmann just used this concept. He wanted to actually figure out that is it the white light, or there is something about its components which makes a green algae like do photosynthesis or proliferate or live. Okay. So why do you think green algae, which is cladophora, is green? Simple answer.
It consists chlorophyll.
It consists chlorophyll. Okay. So what he did is, using a prism, he splitted the white light. Okay, so he took a prism and he took a tube in which there was a solution, and in that solution there is cladophora, these green dots. Okay, they are suspended. Now what he did, he used the white light as a beam, and when it came out of prism, it got scattered into all 7 colors. Do you understand, everyone?
Yes, sir.
So when these color passed through this tube in which this chlorophyll bacteria was, in which this chlorophyll containing green algae is suspended, there were different regions made, right? So it will be violet, indigo, blue, green, yellow, orange, and red. Correct? Vibgyor. All different colors will be there. You understand? So he saw that in this suspension of this aerobic bacteria, so when this cladophora, green algae, so there are 2 things in this. First thing is aerobic bacteria, which is inside it. Second thing is green algae. Now we know that green thing is associated with chlorophyll and green plants and green algae. Algae is a plant only, unicellular, simplest kind of plant, and they do photosynthesis, correct? And we also know sunlight is important. Now Engelmann wanted to figure out, is it the complete sunlight, the white light, that is important, or there is some component of sunlight which is specifically important? Now this experiment was a very intelligent experiment to do. So if there is green algae present everywhere in this suspension, correct? And let's say if only some special component of light is required for photosynthesis, then the green algae present in that compartment will do photosynthesis more. Yes or no?
Yes.
And wherever there will be more photosynthesis, there will be more evolution of oxygen gas. Yes or no?
Yes.
And wherever there will be more oxygen gas, there will be concentration of aerobic bacteria. Yes or no?
Yes, sir.
Right?
Yes.
Yes. So if you can figure out where do these bacteria accumulate, you can figure out where is oxygen being released the most, and you can also figure out which spectrum, which component of the white light is most critically required for photosynthesis. If you see bacteria everywhere in the same concentration, then what will you infer in this experiment? Hypothetically, let's assume that this was the result, that the aerobic bacteria, after shining the white light through a prism on that tube, if you find aerobic bacteria everywhere, what does it mean?
Then all the components of white light are necessary for photosynthesis.
Yes, perfect. But instead what they figured out that it's not all the components are not equally important for photosynthesis. So some components, some regions of white light is more important for photosynthesis. Okay, and he observed that the bacteria got accumulated. So I'll be using, let's use black color for the bacteria. Let's use. So the bacteria got accumulated more majorly in two distinct compartments, okay, which is blue and red. So bluish. So this seeping into a little bit of green as well, or very down here at red. So either in this pocket or in this pocket. Okay, so you can write down the bacteria accumulated mainly in the blue and the red split spectrum. So we figured out, now we all know the wavelength. Now we know the wavelength of light. So we know that the visible light goes from 400 nanometer, which is violet, to 700 nanometer, which is far like red. So somewhere around 550 to 600 nanometer is your blue green component, right? So this you don't have to remember, but just remember that blue and the red spectrum were the two spectrums where the bacteria was found. So it proves that these two light ranges is responsible for photosynthesis more. Now we'll remember this experiment. I will come to it and I will connect it later on. Now, coming from there, is this clear? Are all the early experiments clear to you? Do you have any doubt? No doubts. Okay, so one question still stays. Where does photosynthesis happens? Is it everywhere in the green part of the plant? So site of photosynthesis, where does photosynthesis happens? These people started figuring out in a plant. So can you tell me which organelle in a plant cell is responsible for photosynthesis? We know that photosynthesis, yes, chloroplast. We know that photosynthesis happens in the green parts of the plant, and it's green because it contains chlorophyll, and chlorophyll is a green pigment, and pigments are present inside a plant in green color pigment is present in chloroplast. There are other plasts also. So together all these chloroplast, chromoplast, amyloplast, they come under the broader category of plastids. You all know what are plastids in plants? You have studied it in class 10th, right? Cell organelles. Cell chapter was there, the fundamental unit of life. Was it there in your class 10th?
Yes, sir.
It's there in 11th.
It's there in 11th also. Yes. But oh, sorry, then you must have studied a little bit of it about in class 9th. Plastids,
Yes, sir.
etc.
9th.
Chloroplast, plastids, because 9th repeats in 11th. 10th majorly repeats in detail in class 12th. So what was the site of photosynthesis? It is chloroplasts. They are one type of plastids. Chloro because the word chloro stands for green pigment named chlorophyll. Now, how many of you know that how many chlorophylls are there? How many type of chlorophylls are there in the cell? There are two chlorophylls, chlorophyll A and chlorophyll B. Okay, we'll talk about it later, but just remember this here. Now, if I ask you in 11th only, when you are doing the cell chapter, there you studied the structure of chloroplast, right? That's why I believe that we should teach the cell chapter first. That's where I began from. So you know that what is special about plastids or chloroplast? There is something special about plastids and mitochondria.
They're semi-autonomous.
What does it mean?
It means that they have their own DNA as well as ribosome.
Yes, true. Apart from that, what else is there?
Double membrane.
Yes, it has its own membrane system, which is like double membrane. So you know that in a chloroplast, let's try to make a chloroplast here. So there is a, let's say this is a chloroplast. So there is a outer membrane, correct? Followed by a inner membrane. You understand, everyone? And
Yes, sir.
inside, you know that in chloroplast inside, there are these structures which are present, stacked over one another, right? You know what are these structures?
Thylakoid.
The membrane is thylakoid, but what are these structures?
Granum, sir.
Yes. So they are like this. So if you see here, this thing, this one bunch is called a grana. Okay. And this thing which connects two grana, 1 and 2 together, is called stroma. Stroma lamella. And the membrane of this grana is called thylakoid membrane. So you see this membrane here, this membrane is called thylakoid membrane. Is it clear, everyone?
Yes, sir.
This is outer membrane and this is inner membrane. The space present in between this one, this space is called stroma, and this is called stroma lamella. And inside the stroma, you will find small organelles called ribosomes. As you said, they have their own ribosomes and they have their own lipid vesicles or lipid droplets. They also have their own. What is produced? What is happening here? If photosynthesis is happening in chloroplast, what will be present inside a chloroplast? What is the end product of photosynthesis?
Glucose.
Yes, starch. Can I say starch or carbohydrate? Glucose is also starch or carbohydrate only, right?
Yes, sir.
So we have big starch granules, carbohydrate granules. Okay. Is this clear? This is a diagrammatic representation of a simplified diagram of a chloroplast. Now, if you look at this diagram, and if I ask you, where in this diagram do you think photosynthesis is happening? Where? It is happening in this grana and stroma lamellae here. let me, it's happening here, okay, inside this. And the membrane that's covering this is known as thylakoid membrane. So write down the thylakoid membrane of the stroma lamellae and grana. The thylakoid membrane of the stroma lamellae and grana of the chloroplasts is the site for photosynthesis. This question was asked in the pre-medical test. So if the word thylakoid membrane is not there, then you would look for stroma lamellae. If even that word is not there, then you will look for grana membrane. Okay. And if even that word is not there, then you will look for the chloroplast itself. Make sense, everyone?
Yes, sir.
Yeah. Okay, cool. Now, in the stroma, what is happening is these are biochemical reactions that are happening. Okay, we call it enzymatic biochemical reaction by which we are synthesizing a sugar, which is glucose. When we are synthesizing multiple sugar molecules, the cell utilizes whatever it needs to utilize, and whatever is spared, it has to store it. So glucose is stacked with more and more glucose in a long chain, and together it is formed a starch. Okay, glucose is converted into starch and is stored. Long chains of glucose are stored. Okay. Now there are two types of reactions happening in the synthesis of sugar. So write down. Photosynthesis consists of two types of biochemical enzymatic reactions. Sorry. Photosynthesis consists of two types of enzymatic reactions. The first type is light reactions, and the second type is called dark reactions. Now, by the word, you might be thinking that, oh, it's simple. So light reactions happen during the light when there is day, and dark reactions happens during the night when there is darkness. Correct? Yes or no?
Yes, sir.
Okay. So that's not entirely true, but half of it is true. It's true that light reactions depend on direct light. Okay, so they are dependent on light, they are light driven. That's why they are called light reactions. They are photochemical reactions. The word photo means light. Okay, makes sense? Photochemical, light driven chemical reactions. But the dark reactions, it's not like they happen in the dark only. It's just that they are not directly light driven. They do not depend on light, but they depend on the products of light reaction. So when light reaction happens during the day, it's not like that during the daytime, the dark reactions are not happening. They are also happening. But when it is night, in the night time, the light reactions cannot happen fresh because there is no photo available, no light available. So there cannot be a photochemical reaction. But the dark reactions still can continue because throughout the day, light reactions has happened, and the products of light reactions, which are ATP and NADPH, these two are energy molecules in the cell. They have been synthesized and they have been stored in the cell. Do you understand? So dark reactions are independent of light. They can also happen in the presence of light, and they can also happen in the absence of light. Do you understand what is the twist here? Light reactions are light dependent, but dark reactions are not dark dependent. They are light independent. Make sense, everyone?
Yes, sir.
Yes, sir.
So, but just when these reactions were being named, scientists have already named and figured out the light reactions. So they thought the other one are opposite and must be happening in the dark. So they called them dark reactions, only to figure out that they do not only happen in the dark. They are just not dependent on light. So write down. I'll dictate and you write down. The membrane system of the chloroplast. The membrane system, in the bracket write down thylakoid membrane. The membrane system of the chloroplast is responsible for trapping the light energy, and convert it into chemical energy. And that chemical energy is ATP and NADPH, okay? So trap the light energy and convert it into chemical energy. This part is called photochemical reaction. They are called photochemical reactions. And since these reactions, so you can only convert light energy into a chemical energy if light energy is present. Yes or no?
Yes,
Do you
sir.
understand? Does it make sense? Hiram, Devangi, and Yusuf?
Confusing,
Yes,
but
sir.
makes sense.
Sorry, it's confusing, Yusuf. I can.
No, I mean, it makes sense, but it's confusing a little bit.
Okay, so you can ask me to repeat, not a problem. So see what's happening. What is the first step? We can repeat any number of times. That you just have to say, okay? If you don't understand, it's okay. These things are slightly confusing to begin with, but if your foundations are shaky, if you allow me to just move forward, then the time is not going to rewind back. So your concepts will be weak even after I have taught them. Okay, so be judicial. You are learning from me. You are investing capital in it, so you better utilize it. Ask me any number of times. Okay, Yusuf? No
All right.
hesitations.
All right.
So I am saying that for photosynthesis to happen, first question was, what is the site of photosynthesis? For that we understood this part, chloroplast. Okay, I told you that there are chloroplasts, it contains green pigment known as chlorophyll. There are two types of chlorophyll A and chlorophyll B. And we understood the diagram of a chloroplast. Chloroplast have an inner and outer membrane. We don't worry about that. Inside they have stacks of coins. So these coins, each coin, can you see that these look like coins stacked together? These one? Can you see the cursor?
Yes, sir.
So they look like coins stacked together, right? Each coin is called a grana. So together it forms the grana. Now, two stacks are connected by a similar long extension called stroma lamellae, and the matrix is called stroma. Then you have ribosomes, lipids, and starch granules present in the chloroplast. Now, this stroma lamellae and this grana, they also have a membrane, no? They will be having a boundary. A coin has a boundary, right? So that boundary is called thylakoid membrane. Do you understand, Yusuf?
Yes, sir.
Now on this membrane system, the thylakoid membrane system, first thing that is happening is light is falling on this membrane, and this membrane is converting the light energy into chemical energy, which can be used for chemical reactions, biochemical reactions in the cell. Make sense to you?
Yes, sir.
Yes. So the first set of reactions are called light reactions because they are dependent on light. And that reaction is conversion of light energy into chemical energy inside the cell, into a molecule. So you put that energy into a molecule. Now what's happening is light is falling on the thylakoid membrane. It's converting it into chemical energy. Now, can you convert light energy into chemical energy in the night time?
No, sir.
No, why? Why? So if you have an electric heater in your room and you're living in winters, what do you need to heat your room? You have the heater. So why can't you heat the room anytime? What are you dependent on? Electricity. Yes, because you can only convert electrical energy into heat energy through that electrical heater. Correct?
Correct, sir.
So when can you heat the room? Only when there is electricity. So you are dependent on electricity to heat the room. Correct?
Yes, sir.
Similarly, in light reactions, light energy is converted to chemical energy. So what it is dependent on? Presence of light, sunlight. So it can only happen during the daytime. That's why these are called light reactions, because they are directly sunlight driven reactions, also known as photochemical reactions. Is it clear? Is this part clear?
Got it, sir.
Perfect. Now half of the work is done. We now have chemical molecules which are ATP and NADPH. So these are write down as a result of light reactions. As a result of light reactions, ATP and NADPH. ATP and NADPH is synthesized in the cell. ATP and NADPH is synthesized in the cell. And both these ATP and NADPH are energy molecules having energy in the form of bonds, chemical bonds. But where did this energy came from? Yusuf. Where did the energy stored in ATP and NADPH molecules came from?
Light reaction.
Yes, sunlight. You can simply say it came from sunlight, right?
All right, sir.
Yes. Now the dark reactions just utilize these molecules and do the further chemical reactions. Now they are independent of sunlight, but they are dependent on the products of this light reaction, which is ATP and NADPH, right? So dark reaction can happen any given time. They are happening during the day also, and they are happening during the night also. Write down. The other chemical reactions in the cell, the other chemical reactions in the cell are not directly light driven, are not directly light driven, but are dependent on the products of light reaction. But are dependent on the products of light reaction, which is ATP and NADPH, right? So you can write in the bracket ATP and NADPH. Therefore, to distinguish these reactions from the light reactions, they are called as dark reactions. Therefore, to distinguish these reactions from light reactions, they are called the dark reactions. And last point, this however does not mean, this however Am I audible, people?
Yes, sir.
Yeah, sorry, I just dropped out of the network. It's problematic. Okay. So what I was saying, yes, this, so I was talking about the dark reactions, right? So I said that this, however, does not mean that the dark reaction occur in the darkness. Okay. So they can happen during the day or during the night because they depend on light indirectly through the products of light reaction. They do not directly, they are not directly driven by light. Make sense, Yusuf?
Yes, sir.
Any doubts? No doubt
sir.
at all.
No, sir.
Perfect. So these are the two kind of reactions that happen in photosynthesis. Okay, I will again come back to these reactions. Okay, what is light reaction and what is dark reaction in detail, but this is understood now. Remember, we left this chlorophyll A and B part.
Yes,
There are
sir.
two chlorophyll. Now let's come to this part. So how many pigments are there in photosynthesis? So this part we are going to study about the pigments. So the overall process is clear to you? Pigments involved in photosynthesis. Now I'll also give you the fundamental for this, and then we will go into the details of the reactions, light, dark, and overall what is happening. So as you know, there are different kind of pigments which are present and which are also playing role in photosynthesis, not just the green. So the first is of course chlorophyll A. Chlorophyll A, or I should use small a, it is a notation. And this is either bright green or bluish green in color. Okay. Second is chlorophyll B. Chlorophyll B is yellowish green in color. Okay. And remember, in that experiment done by T.W. Engelmann, which were the two regions where aerobic bacteria were concentrated? The greenish bluish region and
Red.
the reddish region, right? So other type of pigments, third is xanthophylls. Xanthophylls absorb energy from yellowish spectrum. Okay. And the four is carotenoids. What is the first thing that comes to your head when you hear the word carotenoids?
Carrots.
Carrots. You know why? Because carrots contain carotenoids. They are yellowish orange. They absorb energy from yellowish orange spectrum. Now, if you mix green or blue with orange, you will make reddish, correct? Wavelength. So do you understand? It's either towards the green or towards the orangish or reddish. because of these 4 pigments which play a role in photosynthesis. They are present in leaves. Make sense?
Yes.
Now, do you all of you have NCERT with you right now?
Yes, sir.
Yes. Please open your NCERT. We will do a quick in-class assignment. You will do it right now with me. So open your NCERTs quickly. Come to page number 210. Did you open? Can you see there is a figure with 3 graphs in it? In the figure. Can you all see that figure?
Yes,
I
sir.
think it's page 210 for all of us. Now these 3 graphs say something. Okay. First graph, look at first graph. It talks about absorbance of light by different chloroplast pigments. So they have used chlorophyll B, carotenoids, and chlorophyll A. Xanthophylls and carotenoids, they sometimes mix and just show one graph. Second graph talks about rate of photosynthesis, which is measured by oxygen released. And the final graph has 2 things combined: rate of photosynthesis along with absorption of light. Okay, absorption of light. Now I give you all of you 5 minutes by clock. You have to look at this graph and both the x and the y axis you have to read and you have to explain me. So first graph will be explained by Devangi, second by Iram, and third by Yusuf. Is it clear? But to understand each, you have to understand all the 3. That's the trick. Is it clear, everyone, right now? Yes, no, something? Okay, so your time starts now. If you want to read, you can also read the text if it helps you. But exactly after 5 minutes, you have to make notes. I'll be asking you to explain this graph one by one. Your time starts now. Okay, give me a thumbs up if you have started. Because I have started the clock. Cool. Okay, the time is up, people. So ready? All of you understood the graph properly? So the graph is in front of your screen. Can you all done?
Yes, sir.
Okay. So let's start with Divyangi. So Divyangi, first graph. Let's start with this one first, second, and third, and then we will club it all. So what's happening in this first graph? Will you take us through the graph? So this, remember, this is the y-axis that you have to explain. This is the x-axis that you have to explain, and what's happening with the graph that you have to explain. What information is it giving us? Okay, Devangi, begin.
Sir, so this graph shows the absorbance of light by different chloroplast pigments, and the y-axis shows the rate of absorption, and the x-axis gives the wavelength. So in this graph, we see that
Y,
chlorophyll.
yes, y-axis shows the absorbance level. The
Yes.
total graph talks about the rate. It does not talks about the rate. Rate is shown in the second one here. Here it's absorbance versus wavelength.
Okay, sir.
So let's mark here. This white line here is around 400, right? And it maximum goes till 700. I told you why. Because this is the visible range. So this is 500. This is 600. Yes, now tell me.
So chlorophyll A absorbs the maximum blue light at
This is
around
chlorophyll
4
A.
blue colored line.
Okay. This one, right?
Yes, sir.
Yes. So what does chlorophyll A absorbs?
Blue light.
Only blue light? How many peaks do you see?
Bright green and bluish green.
Just because it is blue in color, don't go with the color. Just look at the peaks. Tell me about the wavelength at which you see the peak. How many peaks do you see in this graph for chlorophyll A?
2 peaks.
Yes, one is this peak, right?
Yes, sir.
Which comes around somewhere around, let's say 410, 400 to 4 and 400 to 420. This peak is approximately around 410 nanometer. Yes. The other peak is this, right?
Yes, sir.
And where does this comes?
Around 660.
Let's say around 660, 670, 680. Let's say 680, close to 700. So what is this region? 400. If we start with the Vibgyor, what is this region? It starts with bluish light, right? So this region is more towards bluish region, violet, indigo, blue. And this is towards
Red.
reddish region. Now, do you understand and realize now from this graph why when T. Engelmann did this experiment, he found aerobic bacteria at two different regions, concentrated at two different regions?
Yes, sir.
Because chlorophyll A, which is the most predominant pigment responsible for photosynthesis, absorbs the highest in bluish region and then after that it absorbs energy in the reddish region. Make sense, everyone?
Yes, sir.
Yusuf, is it clear to you? Crystal clear?
Yes,
That's
sir.
why T. Engelmann figured out 2 regions where this bacteria were located. Okay, so I told for chlorophyll A. Now, Devangi, what else? There are 2 other lines. What are they telling you?
So that chlorophyll B
This graph, by the
absorbs.
way, comes directly sometimes in the exams to speculate and infer in entrance also and in boards also. Yes, continue.
Chlorophyll B absorbs the maximum light at around 460 nanometers.
Maximum wavelength energy at around this, somewhere around 4. Let's say, okay. So this is around 460, which is still what light? Bluish light only? Slightly moving towards green now, but bluish,
Yes.
right? Yeah, but predominantly it's bluish. And does chlorophyll B also have a second peak?
Yes, sir.
Where it is?
Around 650.
This one, right?
Yes, sir.
Very close to chlorophyll A. So you said around 650, let's say that. Yes. So this is again towards the reddish region.
Yes, sir.
Another reason why bluish and reddish areas were more aerobic bacteria were present. Now, carotenoids. One peak of carotenoid is very close to chlorophyll B, right? Which is just here, correct? Around
Yes,
450,
sir.
let's say. Again, bluish region. The other peak is also close to 460 only. Let's say this time it's 480. Can I say 480?
Yes, sir.
Still bluish, right? So carotenoids have one peak around 450 and one around 480. 2 peaks. And so either bluish or reddish. You see that absorbance is maximum only in these 2 wavelengths of light. Clear, everyone? So if the question comes, photosynthetic pigments absorb the most energy in which 2 wavelength range? So first is 410 to 480. This is the range, nanometer. And which is this range? This is the bluish range, right? Bluish greenish range. Yes or no?
Yes, sir.
So this information we got from this graph. And what is the second range in which they absorb? 650 to 680 nanometer, which is this range. Which is this range? Reddish range. So this information comes from graph number 1. Is it clear, everyone?
Yes, sir.
Iram, graph number 2. What does this tell you about? Are you there, Iram?
Yes, sir, just give me a second.
Yeah.
Yes, sir. The second graph depicts the wavelength at which the maximum photosynthesis occurs.
Maxima of photosynthesis depicts the wavelength. Okay. What is the y-axis and what is the x-axis here?
The y-axis is the rate of
Y-axis is rate of photosynthesis.
Sorry, sir, there's a little disturbance. Sorry.
No, it's fine. We all are at home, right? So that's That's work from home life.
Yeah.
Yeah, it's the new
Yeah.
normal. Don't worry, you don't have to be sorry. I'm also at home, recently someone comes to the apartment, you have to open the door, get water, etc, etc. Yeah, cool. Yes, so
Yeah.
what is y and
So,
what is x?
so the y-axis depicts the rate of photosynthesis, and the
What
x
do,
is the
yes,
wavelength.
what do we mean by rate of photosynthesis?
I mean, when it get when the graph shows peak and that one.
Forget about graph. What is rate of photosynthesis? What is the rate of your heartbeat? Anything over time is called rate. Remember that. Anything over time is called rate. Okay. So if your heartbeat is 72 beats per minute, that is your baseline rate. Your rate will increase if it is increased by 100 beats per minute, or let's say 110 beats per minute. Similarly, the rate of photosynthesis is measured by how much oxygen is released per minute or per hour or per second time. Okay. Will you remember this, everyone?
Yes, sir.
Yeah. Okay, Iram. So y-axis is clear. What is x-axis?
X-axis is the wavelength.
Yes, it is same. 400, 500,
600,
600, and
700.
700 in nanometers. Yes. And what does this graph tell us? What is this black line going like a snake, making two mountains? Iram, are you
It
there,
is
Iram?
the rate of so it
Yeah.
is the rate of photosynthesis. It shows the rate of photosynthesis, and that's quite similar to the absorption of chlorophyll A pigment.
Yeah, that you don't, there is no absorption here. So let's go one step at a time. You just give the information which is present in this graph. I think this has to be presented by Yusuf. So don't make his work easier and leave your work. You have to present your work first.
Um,
Here is no information about absorption at all. It is, what is this black line telling you? So what I'm marking three regions, let's say region A, region B, and region C. What does this signify? Two are mountains, one is trough and crest. Sometimes reading simple graph is not that simple, right? Are you there, Iram? Because you're
yes,
mute.
sir.
So what does A, B, and C means? Which is the highest? Let's draw a line.
A and C is the highest,
Both cannot be
which
the
is
highest, no? If I draw this, I think this is higher than C or somewhere similar. Okay, let's say A is the first highest, C is the second highest, and B is the lowest. So what does A signifies? If I go
uh,
from like this, it
A
intersects
is
here.
in,
So what does it signify? And it intersects here.
it is around 400 to 450. Yeah,
Let's
it
say
is
it's around 420, I can say 430, let's say. So around 430 nanometers. What does it tells?
It tells that the maximum absorption is around the blue
There is
region, near the
no
blue region.
absorption in this graph. No, not a single word which is absorption. Where is absorption in this graph?
I mean the rate of photosynthesis.
Yes. This graph tells that at around 430 nanometer of wavelength, when you give a plant 430 nanometer wavelength of energy, light energy, it shows the maximum rate of photosynthesis. And then the second highest, it shows at this point when you give it around 680 nanometer of wavelength. So there are two peaks for maximum rate of photosynthesis, which is measured by oxygen release. So you just figure out how much oxygen is released and you can say, and the minimum is a minimum also, which is around, let's say 580 nanometer. It's the minimum because the oxygen released is the minimum. So when you give a plant a light energy of wavelength 580 nanometers, it performs very poor in photosynthesis. It does not do photosynthesis properly. Is it clear? Everyone, is this clear? This is what this graph tells you. Yes, no, thumbs up, something.
Yes.
Yes. Third graph, Yusuf, your turn.
Yes, sir.
What does the third graph tells you? Start with y-axis and x-axis.
The x-axis says about the wavelength of light in nanometers,
Yes.
and y-axis says us about the light observed.
Absorbed, not observed.
Yep.
Observed and absorbed are two different things.
Okay. So these graphs together show that most of the photosynthesis takes place in the blue and red regions of the spectrum.
This graph, you are just speculating it. So if a question comes on this graph in entrance, you all will choose up a wrong answer. This graph tells you that there is a correlation between rate of photosynthesis and the absorption of light energy by chlorophylls, the pigments. So they have taken, what does this blue line signifies here, Yusuf?
Absorption.
Of what? What is absorbing what? Absorption is a word, it's a term. It is not science. What is absorbing here and what is being absorbed? Can you see this blue line is a copy paste thing of some other blue line somewhere in the graph? This blue line is the line for chlorophyll A taken from here. Can you see that?
Yes, sir.
So they have taken chlorophyll A as a representative, and these maxima are the absorption maxima of chlorophyll A, which is around, let's say, here it is around at 410 and here it is around at 680. And we see that the maxima of rate of photosynthesis also is around the same region, somewhere around 420, 430, and here also it is around 680, 690. Make sense?
Yes, sir.
So this graph tells that there is a very strong positive correlation between the rate of photosynthesis, where the oxygen which is released is the highest, and the absorption by the photosynthetic pigments. So this tells that at these two light regions, at this region, which is the reddish region, and in this zone, which is the bluish zone, the photosynthesis is maximum, proved with data. Is it clear, all of you? This is
Yes,
how we
sir.
read a graph and infer exactly what the graph tells. So how the kind of questions that I have seen on these three graphs is sometimes they just give the middle graph and ask you, what do you understand from what data can be inferred from this graph? So it is just telling you about rate of photosynthesis. It is not at all telling you about absorption or any other pigments property. First graph is just telling you about pigment absorption properties. It is not telling you anything about photosynthesis. When you club these two graphs in the third graph, then it is making a correlation between rate of photosynthesis and absorption by the pigments, taking chlorophyll A as a representative pigment. Is it clear, everyone? So we don't have to
Yes,
underread.
sir.
We also don't have to overread the data in science. Okay, cool. So I'll stop here for today. And there's still a couple of minutes. If you want to ask any questions, I'll take questions now. And in the next class, we will go into the detail of light reaction, dark reaction, electron transport chain, etc. So next class will be some chemistry, biochemistry heavy. So I will suggest you read the NCERT, brush it up, and come prepared for the next class. Do you have any questions? Anyone that you want to ask?
Sir,
Yes, Yusuf.
can you explain the second graph again?
Yes. So second graph, the black line just tells you about y-axis is rate of photosynthesis. So a different wavelength. Let's say, let me make different points. I'm making red points on this black line. So this study was done like this. Many different red points were made. Okay. Each red point is how much oxygen is released at this wavelength. When you give this wavelength of light to a plant, they checked how much oxygen is released. So on the y-axis, there must be some parameter 10, 20, 30, 40, 50, 60, 100. So they pointed all these dots and then they make a curve, and they realized when you give the plant of light energy of 430, around 430, 410 to 430 nanometer wavelength, the plant shows the highest rate of photosynthesis. So the highest amount of oxygen is released from that plant. Whereas when you give 580 nanometer of light, it is the lowest. And then another peak comes at around 680 for the rate of photosynthesis. Is it clear? Yes, sir.
Yes, sir.